Welcome everybody. I think it's 2 minutes past the top of the hour. See people are still kind of rolling in, but I go ahead and get the ball rolling here. So welcome the Arthropod Genomics Symposium X 2025, session number two. In our fifth year already. So hope you enjoyed them in years past, and we have a great one coming up today. Genomic advances in non-model arthropod pests. All the connection information is there in that QR code. So in case you haven't signed up and had this link forwarded to you, please go ahead and register at that because we will send out additional material after the webinars. So with that, this is again our second session. Thanks to Lindsay Perkins, UCARS, for organizing our prior session, session number one that went on April 8th, went off really well. And those videos are up on our YouTube channel, which I'll kind of point that link to you here in the next slide or two. And again, today we have our session two. Very thankful to Pro and David for being the organizers. Hopefully they have their cameras on and have a wave, and much appreciated for all their time and effort. And this is obviously made possible by a lot of people working in the backgrounds, not always going to have their camera on, so they're not want to kind of point out their roles. And again, appreciative of all their support. Kevin Hackett, our national program leader, I guess myself as a lead organizer for the past 5 years, the various sessions I've come through. Brent Operators, moderator, and Lindsay Perkin as well for the prior session. Sorry about that, Lindsay, if you're on. I forgot to list you here for your acknowledgement. Glenn Haynes, who operates our Zoom and does the registration pages, and also edits the recordings that go up on YouTube, and nothing could go on without Glenn's help and support. Anna Childers, who designed and puts all the information onto the website and kind of gets information out to the X feeds. And Pia, who typically does our recordings, couldn't be here today, so Pro is doing double duty. He's moderating and playing videos. He's just that good. So again, a little bit of the additional information and resources we have out on the web. Again, there's the website link, and I can drop all these into the chat once we get going in case you don't have these available. And again, that registration link, which you haven't signed up for, please do so. Then you will get that direct link and announcement when these recordings go up onto YouTube. Again, we have the direct link for that there. And we do have a Slack channel where you can request admittance to a Slack channel. So that provides a link right there for doing so. And again, check out our playlist that will be coming very shortly. And last but definitely not least, thank you to all of our presenters. And this time I'm going to turn it over to our organizers, Pro and David. And put one last reminder during the webinar, please do not ask questions into the chat, but there is a Q&A function. If you look on the very bottom center of your screen, click in that and you can ask the questions, and then the presenters can respond directly back to that question. It keeps things a lot more organized and kind of able to respond back appropriately to all your questions at that point. So now I'll turn it over to Pro and David. Thank you.
Well, thank you. And so thank you all again for attending our session. So the goal of our session was to be able to demonstrate some of the unique and interesting research opportunities that have been happening in, arthropods, but that are not necessarily models. I know that the previous session kind of touched a little bit on some of the more plant-based ones. And so we have a little bit of everything here splattered, and my personal favorites of ticks and flies. So with that, we have Michelle Johnson, who will be kicking us off. And so I had the pleasure of seeing Michelle's talk at PAG this year. And they did an amazing job of presenting a really fascinating topic. And so with that, I'll kick her off.
I stopped sharing, so you should have all the power. So.
Got it. All right. And I might even share sound correctly this time. All right, let's see.
Hi, my name is Michelle. I'm a research technician with Bruce Hay at Caltech, and I'm here to talk about allele sales, which is a technology we proposed last year, along with our collaborator Maciek Maselko, who's in Australia. And so today I'm going to be talking about gene drive, sort of generally, I'm going to pitch allele sales. I'm going to show some modeling results because I'm the computational biologist for the lab. I do all the modeling. And I'm also going to give some future applications, so sort of where we see this going. So to start off, the lab I'm in, we're interested in genetic population management. So how can we use genetic engineering? How can we use specific genetic elements in order to alter the traits or the fate of a wild population? So one way is through modification. If you have an element that can spread through a population, you can eventually change some trait of a population. We're also interested in suppression. So how can you use these technologies to eliminate a population, whether it be an invasive species, a pest, or some other species? And so the way most people think about doing this is through a technology called self-sustaining gene drive. So self-sustaining gene drive is where an element will increase itself in a population. Once you release it, it can go from 10% frequency to 100%. So the two main ways we think about doing this is first through homing. So in homing an element, for example, just a Cas9, some guide RNAs, an element will, in a heterozygote, cleave the other copy of that area. And through homologous recombination, you will go from being a heterozygote to being a homozygote. And so this element converts heterozygotes to homozygotes and increases itself in a population. Another broad sort of the umbrella of how self-sustaining gene drives are made is through a toxin antidote system. So for example, if you have a female that has both toxin and antidote, and she mates with a wild type male, individuals will either inherit both toxin and antidote and therefore be rescued, or they might only inherit some lingering toxin through maternal deposition, in which case they will die. And so there are a couple examples of this, but these are the ways that gene drive can work in a population. In both of these systems, our self-sustaining transgene, whatever it may be, will be spread to high frequency. And so for biologists, that's really exciting. We can, put a genetic element, put a few transgenic individuals in a population and see, complete takeover. But for releasing gene drives into the wild, this is less ideal, I think, for a couple of reasons. First, there's a lot of public pushback to this concept. Transgenic strategies where, we're converting an entire wild type population doesn't sit well with everyone. There's a lot of fear around permanent ecological change and damage. So, in the past, a lot of different entities, a lot of different countries have tried biocontrol in different systems and hasn't always worked out. And so finally, modeling right now isn't totally sufficient to ensure desired outcomes. Just because the models are pretty certain that a certain thing will work in a certain way doesn't mean that we're totally convinced. So thinking about self-sustaining gene drives and how it's difficult to get them to, pass regulatory barriers. There's a lot of challenges to getting them out into the wild. We were also looking at this statement from Australia's Office of the Gene Technology Regulator, as we were trying to come up with new ways to do gene drive or genetic pest population management. And so this statement from Australia says that individuals that have an editor, so originally they were thinking about zinc-fingered nucleases, I believe, but we're thinking about, Cas9 cutting elements. If you have an editor, an individual with both editor and the edits it creates is considered genetically modified. If it mates with a wild type individual, its offspring that also inherit the editor are considered genetically modified organisms. Those that only inherit the edit, if it's a simple edit, like a base pair switch or mutations, indels caused after non-homologous end joining, those individuals are not considered genetically modified because in theory, those edits may have occurred naturally in the wild. So an edited individual is not considered transgenic if the transgene that did the editing is no longer present. So thinking about this, we came up with an idea called an allele sale, in which an editor, which is the wind, creates viable fertile edits. And so the editor will just hang out at the same frequency in a population, but as it comes into contact with more and more wild type individuals, it will increase the number of edits in the population. And so at its simplest, you just take a Cas9, a guide RNA, you cleave some target allele, and thanks to inaccurate repair from non-homologous end joining, you can make that target loss of function. So in theory, the number of genetically modified organisms in a population stays the same, but your specific trait that you're trying to get into the population will increase over time. So I'm a computational modeler. My job is to look at this idea and say whether or not it'll actually work, whether it's something that is worth pursuing in the lab. So here's what I found. I found that an allele sale introduced at a small frequency can spread edits to high frequency, to near fixation. And so first, we checked to make sure that just an edit by itself behaves the way we expected. So if you have a fitness benefit, if a trait has a fitness benefit, the allele will increase in the population. Alleles with a fitness cost will decrease in the population, and a neutral edit would remain at about the same frequency in a population. When you add an editor, when your individuals have both the editor that causes the edits and the edit itself, the edit goes to fixation. It increases really rapidly. And the editor, as we expected, remains at some stable concentration in the population. We see that for edits that bear a fitness cost, we can still get the edit to fairly high frequency, but eventually it starts coming back down. This is because the editor is eventually being lost in the population. Because even though the editor doesn't bear a fitness cost itself, because it's always in individuals with the edits, it's slowly being removed from the population. And we see that for an edit that has a fitness benefit of some kind, the edit goes up, and the editor, again, gets this little boost from being with other individuals, being in individuals that have a fitness benefit. So that's for modification. Where can we use this? So I'm going to pitch a couple. It seems like there are a fair number of species in which we're identifying specific genetic elements, specific genes that can be associated with fitness traits that would be beneficial in the future. Specifically, we're thinking about anticipatory edits that address some future condition that we can see due to climate change coming that maybe organisms are not yet under selection for. We're also thinking about human impact. So, for example, sensitizing whether it's human disease vectors like mosquitoes, which are the classic individual, or plant disease vectors, if we could sensitize them to an outside stimulus, such as, by breaking up insecticide resistance genes or otherwise inducing some sensitivity, that could be really useful. And possibly there are a lot of disease vectors where we could find edits that stop them from being vectors. A lot of different plant diseases are spread by insects and arthropods. So those are things that we think an allele sale could do for modification, but we're also interested in suppression. So can we decrease a population over time using an allele sale? The way we decided to try and do this is by considering a population where femaleness requires a dominant allele of some kind. So a great example of this is in the Mediterranean fruit fly, Cerathitis capitata. Females have two functioning turned on versions of the gene TRA, short for transformer. In males, there's some gene for maleness that just represses TRA. So TRA on, female, TRA off, male. We figure you could use an allele sale to cleave TRA and make it non-functional, thanks to, again, that non-homologous end joining. And then you could convert females to males in a way that can then be passed on to progeny. So I took some modeling. I wanted to know in an XY system, so that's what the Mediterranean fruit fly is, they're XY. Does it work? Can we release a single introduction of 10% allele cell bearing individuals and see population suppression? We didn't get suppression. We managed to see a little bit of a population decrease before it went back up. But what was interesting is that the edit was increasing. More and more of this transformer gene was being cleaved and becoming loss of function, even as the editor was decreasing over time. What was also really interesting was that the Y chromosome was completely lost in the population. So now, if you'll indulge me, because this is a topic I find really fascinating and love talking about, I want to think about why this happened. So here on the right, I'm showing an equilibrium path of turnover, which is from sex determination system turnover literature. So on the x-axis, we're showing the frequency of a Y chromosome in a population. Sorry, on the x-axis. On the y-axis, I'm showing the frequency of loss of function TRA in a population. So that's the target that we're cleaving with the allele cell. And every point on this line represents a population that had half males, half females, a perfect 1-to-1 ratio. Despite the fact that often they have a mixed sex determination system. Some individuals have Ys, some individuals have loss of function TRA. So we didn't think about, there being two endpoints. So here at the bottom is a population where all individuals have either a Y chromosome or no Y chromosome, and that determines maleness. At the top, we see that having a functional TRA makes you female, having no functional TRA makes you male. And so we've switched from one sort of males being heterogametic system to a different males being to a females being heterogametic system. And as you can see this line, as you increase the frequency of loss of function TRA, the population sort of ends up balancing by reducing the amount of Y chromosome. So that's why with even a signal introduction, we accidentally push this population to the point where the Y chromosome is completely lost, and instead the presence or absence of TRA is sex determining. Okay, thank you for indulging me. We were also curious to see whether multiple releases of an allele cell could cause suppression. So like with SIT, with FS riddle, a single introduction is not enough. You usually need a bunch. And so we see that with this sex system switching allele cell, it works much better than FS riddle. It can collapse in situations where FS riddle does not. And we see that at lower introduction frequencies, the allele cell can work where FS riddle doesn't, and it also seems to work faster. So the allele cell can suppress in an XY system. Our collaborator, Maciek, is more interested in aromatase in, I think, zebrafish and other fish. And so we wanted to know if this also works in a ZW system, just to check. And so for a single introduction, we saw something similar. We saw a population that decreased for a little while and then started increasing again. We saw the edit increased over time, the editor dropped out, and we saw that the W chromosome went up over time, but didn't go all the way up. So we were sitting at a population towards this endpoint that had a W chromosome, a Z chromosome, and our loss of function aromatase in the system, just all being happy. So again, if you'll indulge me, here's that same equilibrium path of turnover I showed before, except now on the x-axis, I'm also showing the frequency of a Z chromosome. And so in this bottom right corner, we can imagine it being a wild type population where presence or absence of Z, or presence or absence of W is what allows you to be female. At the top, all individuals are WW, and it's the presence or absence of aromatase that determines sex. And so we see that this curve is a much different shape. And even if you increase the frequency of our loss of function aromatase up to 50%, or past that up to 70% even, the population can still be at a happy equilibrium with 25% Z chromosome in the population. So that's why in the XY system, we saw total turnover and the Y chromosome was lost. But in this ZW system, there's a much larger, space of happiness. The equilibrium path of turnover is much longer. So it's easier for the Z chromosome to exist in the same population as our loss of function aromatase. Okay, thank you for the indulgence. We went on to think about, again, multiple releases. We did this for both populations where a WW individual is considered viable, which looks super similar to the XY system. And we also did this for cases where WW is maybe non-viable. And we saw that because you're creating more zygotes that will eventually not make it to adulthood, you can collapse the population even further and even faster, and at smaller introduction frequencies. So we're interested in ZW. We think ZW populations might be more exciting for allele cell suppression systems. So what are actual applications for this? Well, the spongy moth is an invasive insect and a pest, and turns out it has sex determining gene. It has a ZW sex system. Its sex determining genes are being investigated, and there's already genome editing occurring in other moths. So I think the spongy moth could be an interesting avenue to pursue. There are also a lot of insects for whom this complete sex reversal has been shown, even if they're not ZW. So I already mentioned Ceratitis capitata, which is XY, but there's also a bunch of other fruit flies, such as Batrocera dorsalis, the Queensland fruit fly, the olive fruit fly. All of those are in the family Batrocera, but there are other fruit flies that are pretty serious pests, such as the European cherry fruit fly. And so I found one example of in Anastropha. This complete sex reversal does not work, but Ceratitis Batrocera, it's possible that a lot of other insects, between those families and in other families such as Ragolitis, it's possible, but just more digging has to be done. That was in the Tephrididae family. And so Mustardae, I think Musca dorsica, which we'll be hearing about later, also has sex reversal been shown. So there could be a whole host of flies and insects for whom the sex reversal is possible and drive could be pursued, but for whom we just don't have that information yet. Arthropods also includes crabs. So it turns out Chinese mitten crabs are a ZW system, and they're quite invasive. And there's also the European green crabs, for whom more information about sexual development is being found. So I think the allele sale for both modification and suppression is interesting and should be pursued. And our collaborator Maciek Maselko is currently doing work in fish for the allele sale, getting lab tests up and running to make sure it works the way we hope it does. But here I just want to throw out a quick caveat about gene drive modeling and how it's being used for risk assessment. So there's been a lot of modeling that's happened as the gene drive field has gotten bigger and a lot more experiments have been done. In 2023, there was this really interesting paper called Leveraging Eco-Evolutionary Models for Gene Drive Risk Assessment, which is specifically what regulators are thinking about before they agree to pursue a gene drive. This was written by Matthew Combs and a whole team of people with the USDA, actually. And so what they show is that while every single model thinks about fitness costs, which is what I just showed you, there are a whole host of ecological parameters that aren't being investigated, from standing genetic diversity to mate choice and mating system and inbreeding to literally like environmental effects. No one was really thinking about this. So just because all the models say something works a certain way, when we're not including all of these really important parameters, it's much harder to tell what could actually happen in the real world. And so since this paper has come out, and just generally in the past year and a half, a lot has been happening in the modeling world. Just this year, I've seen 2 new papers thinking about density dependence and specifically like how do the different ways we think about density dependence affect modeling results. There's a preprint from just 2 months ago talking about mate choice, which is really interesting, and MG Drive 3, which is this really excellent simulation having to do with, it's mosquito specific. They've included now epidemiological parameters, and they're thinking more about malaria transfer from human to human. And their model is really, really excellent, super mosquito specific, really interesting work. So the gene drive modeling field is getting better all the time, is what I want to say. So my final takeaways, gene drive technology continues to advance, and among that, allele sales are pretty cool. They're being developed in new non-model organisms. So specifically the allele cell, we imagine it's pretty simple, well, relatively simple compared to other drive systems. And so hopefully it can be, transferred slightly more easily to all of these other organisms that people are interested in. And also modeling is becoming more risk assessment aligned. So I see a real future for the field of gene drive. And with that, I'd like to thank Bruce Hay, my PI, Patrick Maselko, an incredible PI and collaborator, everyone in my lab who supported this work, and of course, the people who funded us, and you. Thank you so much for having me and inviting me and listening. Thank you so much.
All right. Thank you, Michelle. I thought that was an amazing talk. So there's a question in the Q&A, so we'll just address that. And then if anyone else has any other questions, you can go ahead and either type it in, or I don't know if we have the ability to unmute you and let you ask, but I'll let you address the question or I can read the question in the Q&A, which is in comparing the ZW and XY turnover, your graph has the frequency of Y or Z on the x-axis. Isn't it more appropriate comparison a Y versus W on the x-axis? Because wouldn't that cause both curves on the Y or W to look the same?
Yeah, great question. So in the case of the ZW system, when you're converting females to, oh wait, hold on. Yes, when you're converting females to males, you end up with more W in the population. So yes, I could have flipped the x-axis, but then instead of the curve going from 25 to 0, it would go from 25 to 100%. So the curve would just flip, going from like the left towards the right instead of going from the right up towards the left. But the curve is still like much longer. I chose to show it the way I did because that way you can see like, oh, it's a similar shape, but one curve is obviously very, very long. And so there's just more space there. So that's why I chose to show it the way that I did.
So Perot, if you unmuted me on purpose,
Yeah.
thank you. That answers the question.
So, actually, I went about doing so. So as the questions come through, I'll just try to go through and unmute people in case they have little clarifications to their questions. But I'll mute you back, I guess. Thank you.
I guess I'll ask one of my questions, which is, so how much of the sex determination system do you need to understand in order to implement these sales? Because, and I think you kind of talked about it, and that's actually even leads to the next talk, which is a lot of us are studying these sex determination systems that we don't really fully understand or yet know. And so, for us to deploy them, how much would you say we truly need to know about the system?
Okay, bare minimum, in theory, if you know that you can reverse females to males and that the product of that reversal is fertile, then I think that should be sufficient for something like allele sales or other sex switching gene drives that are out there. So that requires knowing at least a little bit about what genes are required for femaleness or what could possibly be turned on or off. But it seems like there are a fair number of homologs out there, though I'm not totally sure. But in terms of for full ecological release, so with the XY system completely switching, while, me as someone who's interested in gene drive, I feel like, oh, that's probably fine. If you wanted to suppress a population, it doesn't really matter if you end up permanently changing them. I think there are probably regulatory bodies who might feel differently. So in that case, yeah, knowing a bit more about the sex chromosomes, how they transfer, and then, yeah, probably not too much knowledge, but a solid understanding would be beneficial, I think, especially for getting through regulatory phases. Yeah, that's my answer.
So I also had a question, but maybe it's better to hold off until the final discussion and move on to the next talk. How are we on time, Peru?
Yeah, we could do that. Unless Brenda got bumped up, but I didn't know if she just wanted to be on the cool kids side or if she had a question. Cool kids side, got it. Well, thank you, Michelle. Yeah, so at the end of this,
Thanks,
we'll
Michelle. That
have
was
a
great.
final little panel where we'll just ask any additional lingering questions that we might have. And so it'll be everyone. So with that, next up we have Basanta Bista. And so Basanta is a postdoc with Rich Mizell at the University of Houston, and a collaborator of ours. And once again, he's been doing some really cool stuff. And this almost seems like a perfect transition from Michelle's talk, where they're going to talk about the genomic landscape of Musca domestica. So with that, the challenge of sharing the screen again.
Hello all, I am Basanta Bista. I am a postdoctoral fellow at the Mizell Lab at the University of Houston. First of all, I would like to thank you all for the opportunity to talk at the USDA Arthropod Genomics Symposium. While I earned my PhD studying sex chromosome and genome evolution in turtles, today I will be talking about my postdoctoral research in genome and sex chromosome evolution in the common housefly or Musca domestica. Musca domestica, or the common housefly, is a key model for sex chromosome evolution given its highly variable sex determining system. So the sex in Musca domestica is determined by MDMD, a male determining factor that can occur on multiple chromosomes depending on the genotype. The complex polygenic sex determination in Musca domestica is mediated by the male determining factor MDMD, or the M domain, which can be found either on Y or one of the autosomes. The sex is determined by the presence of this M domain, so the Y chromosome is not essential for maleness. Due to the frequent turnovers and relocation of the male determining factor, sex chromosomes in houseflies are young and dynamic, usually harboring very few differences between the X and the Y other than the presence of the M domain. This makes houseflies a good model to study early sex chromosome evolution, as the relocation of the M domain to an autosome essentially creates a new proto-Y. Here you can see an illustration of the karyotype of Musca domestica. We have 5 autosomes and a pair of sex chromosomes. So this is the females and these are the males. And the alphabet next to the numbers represents the homology to the mural elements, or the arms of the Drosophila chromosomes, and the homology to it. So here females have an XX, and the males have a male determining region in the Y. Again, the male determining region can also move to a different chromosome. Here is an example of a 3M male where the MDMD domain has moved to the 3rd chromosome, creating a new proto-X and a proto-Y. Hemmeisel and Scott in a study looked at the percentages of males having the M domain on each chromosome. So they looked at field samples across continents, across different populations, and they calculated the percentages of males having the M domain on each chromosome, and here you can see the most abundant one are the 3M and the YM. So males having the M domain on either the 3 or the Y chromosome are the most common. Generally across multiple population and across continents, the YM population is more abundant towards the north, and towards the south, the 3M population is more abundant. So this is a phylogeny of Musca domestica with some of its sister species within Muscidae and outside of Muscidae. So when we look beyond the Musca domestica, sex determination is still mediated by the M domain, with the ancestral state being the M on the Y chromosome. The karyotype also remains conserved in most cases outside of Muscidae, with 6 chromosome pairs homologous to the Mueller elements A, B, C, D, E, and F in Drosophila. So within Brachycera, the sister families of Muscidae, Calliphoridae, here represented by Lucilla cuprina, and Sacrophagidae, here represented by the gray flesh fly, can especially be informative because they not only seem to have family-specific rate of sex chromosome evolution, these families also diverged from their common ancestor around 50 million years ago. So nearly all bullflies and flesh flies have the ancestral karyotype similar to these species, with 5 autosomes homologous to Mueller elements A, B, C, D, E, and a heterochromatic X and a male-determining locus on the Y chromosome. So within Muscidae, we see combination of derived and ancestral karyotype. So in some cases, there's a derived karyotype where the Y chromosome is lost. So in these cases, either one of these other autosomes has acquired the M domain, and either the F is either lost or a part of the other chromosome. So there are multiple species that have this derived karyotype. So far we know that the Musca domestica follows the ancestral karyotype with 5 distinct chromosomes, each homologous to the Mueller elements or the arms of the Drosophila chromosome, and a heterochromatic X and a ancestral state where the Y is present on the M, or that's what we thought. So what we've done here is we've sequenced the genome assemblies of M. domestica with different sex chromosome genotype. So for example, in M1, the chromosome 1 here is the sex chromosome, in M2, chromosome 2, 3, and so on. We also have the MY genotype where the Y is the sex chromosome which is absent here. So this is a riparian plot. What this plot actually does is track orthologs across different species or different genome assemblies. So basically, if there is a region, for example, if you look at chromosome 4 here in M1 and M2, if there's a region that connects to another region in a different species without any interruption, what it means is not only are the genes within this region preserved, the gene order is preserved as well. So any changes in the gene order will show up as interruptions or reversals. For example, in this case, the gene order has flipped, so this is a reversal and it shows up as reversal. So when we compare D. melanogaster to L. cuprina, you can see that even though there's not a lot of changes across chromosomes, there's not a lot of movement of genes across chromosomes, there is a lot of rearrangements within those chromosomes that you can see with all of these interruptions and reversals that you can see. In the M. domestica MY, the sex chromosome is actually another scaffold, which is the Y chromosome. But that chromosome doesn't really show up here, and I'll talk about that in a bit. So in this case, I've used L. cuprina as a reference. So basically, it is tracking the order of orthologs within Lucilla cuprina across these genome assemblies. So one thing you'll notice is that there is a region of homology between the X in Lucilla coprina and chromosome 4 or mural element F in melanogaster. So this is the preserved karyotype where each of these Mueller element, each of these chromosomes represent a homology to a Mueller element A, B, C, D, E, and F. But if there's certain regions within Lucilla coprila that doesn't exist, or certain genes here that doesn't exist in other species, those regions won't really show up here because we've used Lucilla coprila as the reference. So here I've rearranged some of these. This is the same figure, but in this case, I've assigned N3 as the reference. So in this case, it is basically tracking orthologs and the order of orthologs across different species on the basis of N3. So here, this X chromosome scaffold actually shows up. So this is the X scaffold from M3. Again, M3, the M domain actually is present in chromosome 3. So this particular scaffold is the X scaffold that is autosomal. And when we compare this to this particular scaffold in MY, where this is the Y scaffold. So I confirmed that this is the Y scaffold because I find the M domain sequence here, and there is homology to certain level of homology to the X sequence, but there's also a region that is unaccounted for. Again, same goes with this figure as well. If there's any region in the M3, because we've used M3 as the reference, that is absent, it won't really show up or connect to other regions in the other species. That doesn't mean that genes are necessarily absent from the assembly. What it may mean is the gene has particular genes have moved, or the gene order hasn't been preserved. That's why it doesn't really show up here. So what this tells me is that there is an X chromosome here that is homologous to some region of the Y, and the Y region in the MY, which would be the ancestor Y, contains certain regions that are not accounted for in the X. So this is probably a Y-specific region. So I wanted to focus more on the differences between the X and the Y. So we looked closer at these scaffolds in these two species. So here what I've done is made the riparian plot again, but this time I've used the M3 and the MY. So when we sequenced and assembled these genomes, we also have phased assemblies. So basically there are two haplotypes representing the diploid genome, haplotype 1 and haplotype 2, for each of the M1 and MY assembly. In case of the M3 assembly, haplotype 2 chromosome 3 is the sex chromosome. This particular scaffold has the MD domain as well, so this would be the proto-Y. And these two represents the X or the proto-X, or the chromosome, autosomal chromosome 3 in these haplotypes. Same goes for the MY. We have the two haplotypes. In this case, haplotype 2 has a Y scaffold, the X scaffold that is similar to the X scaffold in the M3, and the primary and haplotype 1 represents the Y, which has this Y-specific erosion. So when I reorient these genome assembly in a particular way, what does shows up is the region that wasn't accounted for, the Y-specific region, has a small region within it which has a gene order that is maintained. So here, if you trace this brown line across the Y scaffold, it connects to a certain region in chromosome 3 in the M3 assembly. What that means is a small region has basically translocated here. It has the genes are conserved, and the gene order is preserved as well. That's why it shows up in this analysis. So what this means is that there is a region in the Y that is homologous to certain regions in chromosome 3, although that region hasn't been lost for chromosome 3, because if it had been lost, there would be an empty space here representing that loss. But because it's not there, that means that this region has those genes as well, and there's no difference in the gene content between chromosome 3 when we compare to primary. The riparian plot does actually in genomic sequences around what is match ortholog. So the information I provide the software, the riparian plots in sequences, and they're located across schools, and the software have those across the genome. I wanted to look at in particular the actual genome sequences and origins. So I look at the Y-specific scaffold from our YM assembly and compare it to our 3M, so that I can compare an autosome X a Y. What I want to do is to look at the origins of sequences that comprise the scaffold here. So this is the X scaffold in a 3M assembly. So this is autosomal. This is the Y in a YM assembly, contains the empty sexual domain. So when we is a circle that actually represents the alignment, genome alignment of these genomes, these lines that from the or the gray block represents an alignment. So when we align genomes together, we don't get end-to-end alignment, we get patchy alignment that may be smaller or larger. So what I did is put in a threshold filter where I disregard any alignments that are less than 2500 base pairs, so that I can look in more detail at the components of the Y assembly or the Y scaffold in the YM assembly. So at 2500 base pair thresholds, where I disregard all the alignments that are less than 2500 base pairs, it looks like YM mostly aligns to the X, but when we put in a much larger threshold, only look at alignments that are greater than 5000 base pairs, we see a trend. We see that this particular region, which corresponds to the Y-specific region in the Y assembly, has a lot of different alignments across many different chromosomes. So you can see that by looking at the color. So when I increase the scaffold, increase the threshold of these alignments, you will see much more clearly in the next slide. So at 7500 base pair threshold, you can see clearly that even though this particular region in the Y is homologous and aligns properly to the X assembly, this particular region has different alignments to different chromosome, and when I put a threshold of 7500 base pairs, those disappear as well. At a 10,000 base pair threshold, those alignments disappear altogether, which means that this particular region aligns really well to the X, but this particular region has no alignments greater than 10,000 base pairs. So any alignments are bits and pieces from other chromosomes that seems to be less than 10,000 base pair in length. So the next thing we looked at is actually the gene content within the scaffold. So basically what I've done is identified genes, annotated the genome assemblies, and identified genes and their origin in Drosophila. So basically I've mapped these genes to particular chromosome or particular mural elements in Drosophila, just to look at the origin of the X and the Y in Musca domestica. So in this case, M3 primary, in which case chromosome 3 is the sex chromosome, which has the M domain, the X scaffold is autosomal. So in the MY primary, the scaffold here is the Y assembly or the Y scaffold because it has the X scaffold, contents of the X scaffold and an additional Y domain as well. And the haplotype 2 of our MY assembly represents the X scaffold. So basically the scaffold without the Y specific region. So that is probably our X scaffold. So what I've looked at is gene content and the origin of those genes in Drosophila melanogaster Mueller elements. So you can basically see all of these 5 major chromosomes you can identify the homology based on the number of genes that maps to particular Mueller elements in Drosophila. The X and the Y, they have really low gene content. That's why they don't really show up in this plot. But when I focus on particularly the X and the Y scaffolds, this is what we see. So the first row are genes within the X, the autosomal X in the 3M primary, the 6 chromosome X in the ABS or the MY haplotype 2, and the Y in the MY primary. So this is the gene content with respect to the Mueller elements in Drosophila. So you can see here, unlike the previous scaffolds, unlike the 5 other scaffolds, it is hard to assign homology based on the excess of a particular gene originating from a particular Mueller element. In the ancestral state, the sex chromosome or the X should have been homologous to Mueller element F or chromosome 4, but here you see that the contents of the genes tell a different story, that there are genes from other scaffolds, and the 4M scaffold is not the genes from the Mueller element 4 is not the ones that is predominantly present in this scaffold. In the second row, I look at the pseudogenes, genes that have been lost from the scaffold. So these are genes that are still identifiable, but in most cases have certain mutations or frame shifts that introduces a stop codon. So these are pseudogenized genes. And again, when I look at their locations in the Drosophila Mueller elements, we see a different picture when we compare it to the other scaffolds. So by this, we can tell that the Y or the X chromosome, the ancestral X chromosome in Musca domestica likely is not a homologue of Mueller element F because it has genes from other chromosomes as well, and it has lost genes from other chromosomes as well. So it doesn't really make sense that they share the same origin. So what this tells us is, even though the chromosome number resembles the ancestral karyotype that you see in the sister species of Musca domestica, it doesn't actually have the ancestral karyotype. The karyotype is derived. It has somehow lost the F. The genes from the F may have moved to other chromosomes as well. And the sex chromosome, the Y, is something entirely different. It is possible that the F has acquired enough mutations, enough changes across its history that it looks very different from the F in these species, but it is also possible that the male determining locus may have translocated into certain other regions, could be possibly a B chromosome, and other genes moved to that chromosome after the linkage with the male determining region. So this puts into question what the ancestral karyotype is for Muscidae and what the actual karyotype we see in the Muscidae family when we look at Musca domestica. So what's next? So we know that there is a temperature decline between M3 and MY. What that means is the proportion of males having MY increases as you go up north, and M3 increases as you go down south. So basically, there is a temperature decline where one particular genotypes behave better compared to others. So these differences, these phenotypic differences must stem from any differences that these scaffolds have within them. For example, differences between the X and the Y, or these X and the Y across species as well. So we're looking at the repeat content. So as soon as a chromosome acquires a sex-determining locus, there's a cascade of events that happens. One of those is the regions become more susceptible to invasions from repeats or transposable elements. So what we're looking to do is compare the content of transposable elements across species. So for example, compare chromosome 2 in M. domestica, where it contains the MD sex determining locus, against the autosomal forms in the other genotypes. We can also compare the two haplotypes within the same scaffold, which within the same genome assembly. So basically what we're doing is comparing the X to the Y to look at any differences. The preliminary results from the repeat contents tells me that there's not a lot of differences when it comes to repeats, and the repeat contents, or the TE content, also depends highly on the quality of the assembly. So in most of these cases, the genome assemblies were acquired from inbred lab strains that have these particular sex chromosomes. So they are inbred, which means that a phased haplotype genome assembly might be difficult to reassemble from two haplotypes that are generally similar across the chromosomes. So the haplotypes we have aren't as good as the primary assembly. So it is difficult to compare between the haplotypes, and the repeat contents look similar. We also know that the gene content is similar across X and Y. So what this means is any differences that results in the phenotypic plasticity when a particular genotype does better than the other in certain regions must stem from some changes or differences within these genes. So it may be certain mutations that they have acquired and things like that. So what we're doing next is looking at the finer details of the protein coding genes within these scaffolds and comparing them to their haplotypes and their homologous counterparts across different genotypes from different populations. Lastly, I would like to thank all the co-authors of this work for their effort in collecting, assembling, and analyzing these genomic resources. I would also like to thank NSF for funding this work, and I would like to thank you all for giving me an opportunity to talk about it. Thank you so much.
All right. Thank you, Basanta. With that, we can do a quick question or two while I prep up the next talk, and then roll into the next one.
So I don't see any other questions, but I did have a question about the sort of autosomization of the X chromosome, right? And obviously dosage compensation seems like something that would be hard to overcome with that, and it seems very fluid. So do you have a sense of how that's working?
Since there's not so these movements of these genes doesn't really cause a lot of gene loss because there hasn't been a lot of time. And since the contents of the Y and the X in the proto Y and the X are similar, that means that there hasn't been a lot of gene loss. So dosage compensation hasn't really come into play yet for some of these.
So is these, but you're going from one copy to two copies, right, on the X. So is the ancestral Y just has sufficient
you
gene
mean
coverage?
the ancestral Y? I guess so. I guess because we don't find an ancestral Y, it's probably the Y has been lost and the X is now twice as much. But again, depends on how degenerate the ancestral was. If there is need for doses compensation at all for the ancestral Y.
So we did just also get another question in from an anonymous attendee. Do you know why the male determining gene moves to the other chromosome sometimes? You said you were looking at TEs next. Do you think that those are involved? I also have this question.
We don't know. So T's in this case, probably not, because only thing that needs to move is the male-determining region. It may be mutations. It may help overcome some of the drawbacks of, I guess, an older sex chromosomes, like gene loss and things like that. So maybe that's why a higher rate of these turnovers for the sex-determining genes may be beneficial.
All right, are we ready for the next one here, Pura?
Yep, let's do it. So up next we have Alex. I'm gonna mispronounce his last name, but as someone who's faced that regularly, Kneubehl, like the prize. So he's gonna give us a little bit of a talk on some of the challenges one is associated with doing tick genomes. And so Alex gets to live a small part of what David and I's daily life is like. And so I'm glad that there's someone else to at least share the trauma. That's maybe the word I want to say. All right, and so with that.
Hello, everyone. Thanks for coming today. My name's Alex Kneubehl. I'm a postdoctoral fellow at Baylor College of Medicine in the Department of Pediatrics, the Division of Tropical Medicine, and in the section of vector biology and microbial pathogens. And today I'm going to talk about the trials, tribulations, and new opportunities that we've encountered along the way to the first argasid tick genome assembly of Ornithodoros turicata. And so today we're going to be talking about ticks. And for some of you, they may know there are three families of ticks: the Ixodidae, or the hard ticks, the Argasidae, or the soft ticks, and then Allele, which are these kind of single species living fossils found in Africa. What we have here in the United States, we have the hard ticks and the soft ticks, and we're going to be talking about these really cool guys today, the argasid soft ticks. So I kind of want to go over a little bit more of what is a soft tick, because I feel like most people are probably familiar with what a hard tick is. And so most people know these from like either Lyme disease or cattle tick fever. But the soft ticks are around. They're a little bit more elusive and secretive, but they get up to mischief and cause problems for humans and animals. So we'll go into a little bit of that today. And so these guys have some interesting biological differences. One of the kind of key differences is that the hard ticks have exposed mouth parts, whereas the soft ticks have the mouth parts are a bit more hidden. The hard ticks have this dorsal scutum or shield that's on the back of them that the soft ticks don't. And then the exoskeleton differences, obviously you have hard ticks, a hard exoskeleton, soft tick, they kind of have this leathery exoskeleton. And biologically, there's some interesting differences too. So the hard tick lifespan's around 3 years, whereas the soft tick can live up to 2 decades. And one of the bigger differences between them is that the soft ticks will go through multiple ovipositions, so they don't feed, mate, and die. The soft ticks can live, in that 20 years, they're laying multiple egg batches. Feeding behavior, the hard ticks are going to go out in quest. They're going to go look for a blood meal. Soft ticks are more nidiculous. They're going to be more buffet style. They're going to be living in the burrows and dens of their host organisms and then feed on them whenever they want. And the hard ticks are going to have a longer feeding duration, so they got to go 5 days. They're going to embed into a host and then, take that blood meal over those 5 days, whereas the soft ticks are kind of in and out, rapid feeders, 5 to 60 minutes. And so these soft ticks are a pest to humans and agricultural animals, and they do this through a variety of different ways. It can either be physical damage, so this is the ear canal of a racehorse, and so this is a soft tick, Atobius magnini, that lives its entire life cycle in the ear canal and causes damage to the ear and can cause deafness. This also occurs in humans. They can cause toxicoses, so this is where the tick bites, and then you have a reaction to the saliva. So this is a Ornithodoros brasilensis bites that cause these ulcers. It can be they can last for quite a while. But I think one of the big things with these ticks that's a big problem is that they can vector disease-causing pathogens. And so soft ticks are typically associated with what's called tick-borne relapsing fever. These are spirochetes, they're bacteria that cause disease in humans and in animals. And then one of the big viruses that they can also transmit is African swine fever virus. This is where USDA is interested because this is particularly can be devastating to the pork industry where the virus is circulating. And so these argasid-borne pathogens, they circulate in complex cycles with the tick. So the ticks are obligate blood feeders. And so you have these pathogens of interest, so this tick can be infected. And so those ticks have to feed on vertebrate hosts. And so the ticks that we work on Ornithodoros turicata are pretty generalist feeders, so they can feed on a bunch of different organisms. And so they can transmit to those hosts, and vice versa. They can also acquire pathogens from the hosts that are out there. And they can sometimes run into incidental hosts, be that an unsuspecting human or a pig on a farm. And this is where you can have kind of these severe consequences for disease. So in humans, it'd be relapsing fever, and in the pigs, the pig farms, it could be African swine fever virus, which is particularly devastating for the pork industry. And what's kind of an interesting feature about argasid ticks is that they can undergo transviral transmission for a number of different pathogens. And so this is essentially vertical transmission of the infected female tick passing on those pathogens to her eggs. And so when the eggs hatch into larvae, those larvae are already ready to go and can start transmitting pathogens right away. And so this is a way that the vector itself acts as a reservoir, and that infected female can start an endemic focus of disease in an area pretty much by herself. And so it's kind of another unique biological feature. And again, remember, these ticks can live up to 10, 20 years, and they're undergoing multiple, ovipositions during their entire life. So they have the capacity to potentially generate a lot of infected ticks if she's passing that particular pathogen transvirally. So a lot of really interesting disease maintenance dynamics that are going on here with these argasid-borne pathogens. And so one of the kind of roadblocks we often run into in our particular area in the argasid research is that we are kind of lacking in those genomic resources. And that's a common problem among probably a lot of the tick groups would say that's a common problem. But we're starting to get better and better about having those hard tick genomic resources. But the argasids are going to lag behind that. And one of the I'm going to go into a couple different areas that we're interested in and where we kind of hit roadblocks by not having that underlying, genomic resources is looking at dispersal and invasion. And so having, population genomics and phylogeography studies to do that, would really help to have some kind of underlying genomic resources to pull from to be able to do those kinds of work so that we could see how these ticks are moving in nature. Our lab does a lot of vector host pathogen interactions. And so, having the genomic resources to overlay with the RNA-seq data and the proteomics data really helps make those data sets, kind of the omics data sets, more powerful because we have, if we have an annotated genome, it's much easier to kind of look at the functional aspects of, the RNA-seq and proteomics instead of trying to do those individually de novo. And then having the functional genomics, different assays that we can do and approaches really helps by having the genomic resources. And then, we sometimes run into issues even looking at basic biology, at least on the molecular characterization side. So, looking at aspects of reproduction, as Job Lopez is going to talk about in the next talk, having the genomic resources really helps make that more feasible. Looking at psilomics, so this is looking at how the saliva is composed at a proteomic level, and then looking at how that can change in different conditions, different interactions with the environment, different interactions with pathogens, in the context of the vector, how the host responds to saliva. So those are things that we could kind of run into issues sometimes without having the genome available. And it's also a problem for systematics, right? So this is, looking, the mitochondrial genomes will get you only so far, but having the nuclear targets as well can also be really helpful to look at the relationships between different tick species and how they've evolved over time. And with all of these different things, different aspects, we run into, kind of these hurdles we have to overcome without having the genomic resources. If we had those resources, that all helps to contribute towards the development of control measures. And so, by a better understanding how they're dispersed in nature, and by having a more complete understanding of vector host pathogen interactions and, the basic biology of the tick, we could kind of start to develop ways to combat both the vector, the pest, and the pathogens that they transmit. And so one of the aspects of the vector biology and microbial pathogens group is to help bridge that gap in genomics resources for argasid ticks. And so we initially started working on mitogenomics because it's a little bit more approachable for the infrastructure that we had. But then when the USDA got involved, we moved that into whole genome sequencing to really build out the infrastructure for argasid ticks. And so we'll kind of talk about the mitochondrial genome stuff first. So it's really useful for the genetic analysis of ticks. So you have the mitochondrial genome. It's also known as the mitogenome. It's this 15 kb replicon that exists at higher levels in the cells. So this is some approachable target for sequencing. It's really helpful in doing species identification and has been, used in systematic analyses for the last 2 decades. It's been kind of foundational to our understanding of tick systematics and tick taxonomy. And it has been used to differentiate interspecies populations. And so people have used this in population genomics and phylogeography studies. But it's currently a little expensive to generate full-length mitogenomes. As you start to scale up, it's not as bad if you have enough money to drop, all at once. But if you only have like 1 or 2 samples, or if you're kind of in the low to mid throughput, it could be kind of expensive. And so that was one of the areas that we wanted to help the community, is to develop a full-length mitogenome PCR and sequencing pipeline that was cheap. And so it's pretty straightforward, using the QIAGEN D&EZ kit for blood and tissue. We did extractions. We developed a PCR based assay where you can generate the entire mitochondrial genome as a single amplicon, but we do that from 2 different spots to account for the different primer sites. And then you can sequence those using Oxford Nanopore Technologies with their rapid kit, using the MinION sequencer. And so this is all very approachable for labs that don't really have a robust genomics infrastructure, or really any genomics infrastructure at all. And the bioinformatics is very easy. You could do it with a laptop. And the nice thing is the cost per sample is around $10. If you're doing 1 sample, it's $15, and you can use the service provider to even do the sequencing for that. So it's very approachable. And so we published this pipeline, and also did kind of stress test the assay with sequencing around 169 samples from all across all 3 tick families from around the world. And so this was an international collaboration, including the USDA. And so one of the focus areas for this was ticks from South America, particularly Ornithodoros rudis. And so this is a tick that morphologically shares characteristics of the Electrobius and Ornithodoros, but there wasn't a lot of mitochondrial genome data that was available. And so doing a protein coding gene analysis from the mitochondrial genomes and phylogenetically, we were able to show, kind of through an integrated taxonomic approach, that yes, Ornithodoros rudis is nested between Electrobius and Ornithodoros, but also really doesn't fall into any of the other previously described subgenera. So it seems to be its own thing. Further, we also looked at Electrobius more. So we fleshed out that subgenus with more of the species that fall within that. And so that helped to show the monopoly of that group. And so that Electrobius was then elevated to a genus status, and so that is now used in tick taxonomy. So we've also used this pipeline in the description of a new tick with Sebastian Munez-Leal, and that was Ornithodoros improvisus from Chile. Also with Sergei Filatov in the lab, we also looked at the phylogeography of Ornithodoros verrucosus, which is found in the Caucasus. and in Eurasia. And so we were able to use the pipeline to do that analysis. And so that's kind of available for the community to use, and it's something we use routinely in the lab. But we still really need a nuclear genome assembly to help investigate, those kind of fundamental questions about argasid tick biology that we have. And so in the beginning, this is around like 5 to 8 years ago now. We had the 10x Illumina Ornithodoros turicata genome assembly, and it really wasn't good and wasn't doing what we needed it to do. And so the total size was around 1.2 gigabases. It was around 77,000 scaffolds, so pretty fragmented, and it had a very inflated number of coding sequences than we were expecting, around 127,000. And so we were expecting many duplications and fragmented genes to be in this. So this assembly was annotated by a private company using an aphid gene model at the time. So that was just what we had available. And so this wasn't really the best. So the Busco score for arachnids was, 89.4%, so it wasn't terrible. But the fragmented gene percentage was pretty high, around 6.7%. So there's a lot of room for improvement here. So in comes the USDA, and so they saved a day and helped us to generate a chromosome resolved genome for Ornithodoros turicata. And so that all came about because of the AgPest 100 initiative, where Ornithodoros turicata was identified as a putative vector of African swine fever virus. And so that would be very devastating if the tick, or if the virus got into the tick population. We know these ticks are long-lived. They can potentially do transviral transmission of the virus. And so it'd be a huge problem if that were to occur in the United States. And so as a way to help potentially mitigate that, having the genome, we could potentially come up with some control measures down the line. And so this was a collaboration with the USDA, and that helped to generate the PacBio-CLR data and Illumina Hi-C data. And then that was a assembly of an individual female that yielded a genome size of around 1.1 gigabases, 378 scaffolds, so far better than the Illumina genome we had before. Using the Hi-C data, we're able to scaffold those further into chromosomes. So we have 10 chromosomes as shown here in this juice box plot. The Busco score was very good, 97.8% completeness, and that fragmented genes was well below 1%. NCBI went ahead and annotated this, and so we have around 36,000 coding sequences, so far better than the 120,000 we had before. And so this genome is now available on GenBank, and the genome report has been accepted as in press in G3. So that is now available to the community. And so one of the first things we wanted to do with our assembly was look for the sex chromosomes, right? So in argasids, the sex determination systems are thought to be XXXY based on some older cytogenetic data, but there hasn't really been anything in the modern era to help confirm that. So what we wanted to do was kind of leverage having our genome assembly to see if we could tease that out. So we sequenced another 3 females and 3 males using Illumina to help determine the X chromosome, again, because this is a female genome, through read mapping. And so looking at the normalized depth across all the different chromosomes, what we would expect for the female, that is the X and the autosomes would be at around a 1X coverage, but that for the males, the autosomes would be at a 1X depth. But because they only have 1 X chromosome, they would have half the coverage or half the depth for that if that was the sex chromosome. But obviously, we're not seeing that here. Across the males and females, everyone had around 1X coverage. So I wanted to take a little bit more of a granular approach and look across each of the chromosomes. What if we did 1 megabase window? What's the depth at each of those points? And so what we teased out was that chromosome 1 had this difference between males and females. And so the males were around 0.5x coverage in the first 90 megabases of chromosome 1, whereas the females had a pretty decently steady 1x coverage in that same region. And so that's really interesting. That indicates that, okay, the males in that area are homozygous, right? So they're haploid in that area. So we call that the male haploid region. And so one of the things we wanted to look at was, okay, what's the variant frequency in that area? And so if we look at the total chromosome 1, variant site frequency is pretty consistent between males and females. And then when you look, kind of break it down by region, so in the haploid region there, we see that the males have a far less variant site frequency compared to the females in that area, but was relatively similar in the rest of the chromosome. So it really definitely indicates that the males are haploid in that area, they're hemizygous, and that they potentially have a full length chromosome and another chromosome 1 that is smaller. And so that kind of doesn't really jive with the conventional thinking of XY, at least as I knew it before we started doing this. But the hypothesis that we end up coming up with is that the sex chromosome system in O. turicata is an early stage XY system that's still recombining along that shared arm of chromosome 1. And what does that actually look like? So in the pre-diverged XY systems, so you start with a proto-X and a proto-Y, where the proto-Y has this male beneficial genes that are no longer, recombining in the same area as the proto-X, but they recombine along the length of the rest of the genome, or the rest of the chromosomes. And then if you fast forward all that, you end up with what we kind of conventionally think of as an XY systems as a Y that is mostly male beneficial genes and an X that's very different from Y, and there's very little, if any, recombining regions. O. turicata seems to be somewhere in the middle where, we still have a large chunk of recombining regions, but they definitely have in the males an area that is no longer present to recombine with the other copy. And so we still need to tease this out a little more, but it's kind of exciting that this is, some of the first foray into sex determination systems and argasids using, sequencing data. And so some key takeaways and future directions from all this: argasid ticks are cool, but they're a public health threat, and it's something we really need to consider pretty seriously, given that, if African swine fever virus gets into the United States, it'd be pretty devastating for the pork industry. Tick mitogenomes can be sequenced cheaply, and they're important for tick systematics as well as other areas too, like rapid identification of tick species and population genetics. So the Ornithodoros turicata genomes, the first chromosome resolved argasid genome assembly, and this is already proven to be very useful in our lab. This helped us to acquire some other USDA funding and to spun off other collaborations and other projects that we've been able to put in because we have this genome. Some more immediate future directions: obviously the turicata sex determination is still unclear, so we need to generate a male genome to help tease that out and investigate that further. And so we have some functional genomics investigations underway that Joe Lopez is going to talk about in the next talk, looking at oogenesis and reproduction, and that's been really helpful having this the Ornithodoros turicata genome to be able to do that. So collectively, I hope this really helps to underpin, that USDA collaborations with academics are important, and the turicata genome wouldn't have happened without a lot of expertise and resources that the USDA has provided. And so with that, I'd like to thank the Vector Biology and Microbial Pathogens Group, also known as the Lopez Lab, Baylor College of Medicine, and our international collaborators for the metagenomics portion of the talk. Really, hats off to Pete Thiel. He's been a huge force for our studying argasid tick biology and helping make the connections between USDA and other groups with us at Baylor College of Medicine, and has been really a big advocate for the Ornithodoros turicata genome project. And again, our collaborators at USDA, it's been great to work with. It's definitely one of my favorite collaborations. I really want to point out the work that Amanda did with really getting the genome assembly to the finish line, and then Perot helped really sink it in too. Also, thanks to Perot for putting up with my 2 a.m. Emails about crazy projects, and he's been a great person to work with. We have trying to get other grants in and other collaborations started, and so it's been great. I really want to thank the AgPest 100 group for reaching out and helping to move this project forward, and for all the resources and expertise that have been offered.
No, Pro, you're muted. So
I just wanted to say thank you again, Alex. I also just wanted to say I appreciate your digital self just like totally correcting me on your name right off the bat. So, well, I'll start to prep the next talk while if there is any questions, feel free to ask.
Yeah, so I don't see any questions, but Alex, that's all great to see. So I'll just say that I happen to know that that male genome, we've got a lot of the sequencing done, so that male genome will be upcoming. And Alex, I'm curious what kind of analyses you might anticipate doing once you get that second genome, if Alex is here.
And he's muted and his camera is off, but
Maybe he stepped away.
He's having issues with his mic. Okay. So you commented that it's been helpful with teasing out a lot of the sex chromosomes, but do we need to unmute you?
No, it shows he still is muted himself. Just hit the mic button on your bar.
Should just be down the bottom left. You're already unmuted.
Computer just straight up did not want him to talk.
There you go.
No, no, this
there you go.
Can you hear me?
Yep.
Yes.
Oh, okay. It's telling me I'm muted.
Oh, and now you're gone again. Oh no.
You can be doubly muted, but
So hey, why don't we if we got the
we'll
next
just move on
talk
to
ready,
the
maybe
next
we'll move to
slide.
that. And Alex, we can maybe get that working and chat afterwards.
All right, and so closing
Thanks,
us
Alex.
off is another another talk about ticks. And so as you saw, Job Lopez, and despite what my partner says, no, it's actually not Rob Lowe. So he is a professor at the Baylor College of Medicine, and so he's going to give us a little following about more of the functional genomics for portions of Ornithodoros turcata. So let me share that.
Hi, I'm Job Lopez. I'm an associate professor at Baylor College of Medicine. I'm in the Department of Pediatrics, Division of Tropical Medicine, and I head up the section of vector biology and pathogenesis. Today, I'm going to talk about Ornithodoros turicata, which is an argasid tick. Specifically, I'm going to talk about Ornithodoros turicata oogenesis, the vitellogenesis pathway, and really why we care about that and how we've used the tick genome to help us identify key players in vitellogenesis. I think Alex did a really good job of introducing Ornithodoros turicata and the tick genome, so I'm not going to go into the trials and tribulations over the last 10 years with that. What I'm going to do is just jump into some argasid biology, some pathogens that are transmitted, why we care about the vitellogenesis pathway, and then what we've been doing to understand that pathway. So let's first talk about some argasid tick biology because it's a lot different than exodid ticks. So on the left here is an egg that hatches into an unfed larva. That larva requires a blood meal to become an engorged larva and then molt into an unfed nymph. That nymph will feed again on a host and become an engorged nymph. But this is where it becomes different for argasids than exodid ticks. That engorged nymph will molt into a second stage nymph. And so argasids typically have multiple instar stages, as opposed to exodid ticks that'll have one nymphal stage that then molts into an adult. On the last molt, that nymph will molt into an unfed adult, either a male or female. I want to point out that during the process of blood feeding from larva to nymph, we do not understand or we do not know the sex of the tick at this point. It's only when they molt and then we can see the genital aperture it's developed at that point into a male or female is when we can biologically sex these ticks. Okay, so that unfed adult then will feed into an engorged adult. If this tick is an inseminated female tick, it will oviposit and lay eggs. And here comes then another difference between exodid and argasids. That female tick after oviposition does not die. She digests the blood meal becomes an unfed adult that can then subsequently feed and oviposit again. So argasid ticks, the females have multiple gonotrophic cycles versus one gonotrophic cycle for exotid ticks. So let's talk about the bacterial and viral pathogens transmitted by argasid or soft ticks. The first I just want to quickly touch on is relapsing fevers in the genus Borrelia. Depending on the species, these have human, veterinary, or agricultural significance. They cause fever, hence the name, nausea, neurological symptoms, depending on the species. And then in veterinary animals, agricultural, human, miscarriage is a huge problem in pregnant females. The next one I want to touch on is the tick-borne encephalitis virus complex, flaviviruses. So these are hemorrhagic fever viruses of humans. But what I really want to focus on, especially for this audience, is African swine fever virus. This is a hemorrhagic fever virus in domestic pigs. So there are two hosts, just given the life cycle of the soft tick, all of those nymphal stages. What I did not mention in my last slide was that tick life cycle can go on for decades. These ticks are really long-lived. They can oviposit. The female ticks can oviposit and lay eggs. I mean, in the lab, we're going over 5 to 10 ovipositions. So with vertical transmission, the soft tick is a really good reservoir, we believe, for certain pathogens. Wild pigs are also a good reservoir. They have varying degrees of pathogenicity. So some will be completely asymptomatic, while others will have varying degrees of symptoms. But what happens is the tick can transmit to a wild animal, and then there is horizontal transmission of those wild animals come in contact with domestic. And then in the domestic pig population, it's pretty much 100% lethal. And that's what this study in Africa is showing, where different degrees or different images of the different types of hemorrhaging, and how they contain these outbreaks is by culling the population of pigs. So this is a significant concern agriculturally, especially if it were to emerge here in Texas or in the United States. So Texas is, I think, a really good ecological environment for this virus to emerge. These are some pictures back in 2015. We would go down to Chaparral Wildlife Management area and do field work. And I remember one night we were collecting ticks down there, and the wildlife biologist was just showing us pictures, and it was a picture of this kid holding an African warthog he had shot. And I had asked him, do you know what these do, or do you know what these are involved with pathogens? And he wasn't aware at the time, but what he had said was that these were brought in a neighboring hunting ranch. They had escaped, and then they had started reproducing. And Chaparral Wildlife Management Area was one of the first areas where they started invading, because it was adjacent to the hunting ranch. And since then, they have been spreading throughout regions of Texas. So we have the warthog. We published, or let me step back why that matters. So in 1987, Hess published a paper looking at several tick species in the Americas: Ornithodoros porridicensis, Coriaceae, Parkeri, and turicata. And this column shows the distribution. And what they wanted to point out are some of these notable findings, because each of these ticks had different degrees of maintaining that virus. So porridicensis, they really focused, I think the study was very well, thoroughly done on that species. And then with the other species, it was not as thorough. So they showed transstadial and transovarial transmission of African swine fever virus. The ticks could acquire the virus from an infected animal, and subsequently they could transmit it. Coriaceus, they showed transstadial transmission, and they were able to maintain that virus for almost 1.5 years. They also could transmit it to domestic pigs. Parkeri could acquire it, can transstadially transmit it between developmental stages of ticks, but they were not showing infection to domestic pigs. And I think Ornithodoros turicata was really interesting. Unfortunately, this was the tick species that did the least amount of work, but what they showed was transmission was dependent on the colony they used. They had two different populations of tick, and they showed one can acquire it and transmit it to domestic pigs. And they said in the paper this was a preliminary study with turicata, but then I never could find a more in-depth study looking at vertical transmission. So we know in some species of tick, at least, African swine fever virus is vertically transmitted. Other pathogens are vertically transmitted in argasid ticks, like the relapsing fever Borrelia. So we're very interested in vertical transmission. So if we look at turicata, though, in Texas, we have the pigs here, we have the tick, but what we showed in this species distribution model of Ornithodoros turicata was like Texas looks like it has a really good habitat for the tick. So each black dot is where a tick was collected, or there was a record of a tick collection. And then we overlaid these with different environmental variables to generate this heat map of showing suitable habitat for turicata. So yeah, so we have the tick, we have the warthogs, we have a huge feral pig population. So we have everything in place if the virus were to be introduced through Mexico or through the Caribbean. So that's then where we have really become interested in this system. So let's really quickly touch on our section, the vector biology and pathogenesis section, where we really want to understand how pathogens are maintained. So we have three sections that we focus on: genomics and genetics and diagnostics, ecology and vector biology, and animal development. And so what I'm going to focus on is our genomics and genetics section. So this is the core of our lab. Serhi is a postdoc. Michael is now an assistant professor in our section. Alex, the work I'm showing today is Alex. He's a postdoc, and then Brittany is a staff scientist in my lab. So let's talk about egg development and vitellogenesis. So egg development, I think, is important to point out that it can be triggered two ways. Non-autogenously, so this is what typically happens in exoded ticks. So an exoded tick has to mate and then blood feed for oviposition or oogenesis to occur. Egg development will not occur without those. I think the soft ticks are really interesting because they can actually reproduce, and like oogenesis can be triggered solely by mating. So a female tick does not need that blood meal to oviposit and for oogenesis to get triggered. I think this has implications, especially when you think about the establishment of an endemic focus. If a female is introduced, you don't even need a blood meal source for an endemic focus to be established if that female is inseminated and infected. And then once a blood meal host is introduced, that spot then can quickly expand. So oogenesis then, let's talk about that a little bit more. It's characterized by the vitellogenesis pathway. So what is vitellogenesis? Vitellogenins are precursor yolk proteins. They're large subunits produced in the fat body, undergo post-translational modification, and are translocated into oocytes. They're broken down and make yolk. I have a picture I'm about to show that really summarizes this a lot better than just all of these words. So I just want to introduce it here. In exoded female ticks, blood feeding is what upregulates the genes responsible for vitellogenesis. And then in argasid, since they can reproduce autogeneously and non-autogeneously, mating alone can induce oocyte development in vitellogenesis, while blood feeding further upregulates this. And then why we're interested in the vitellogenesis pathways and some agricultural pathogens and some plant pathogens, vector-borne pathogens, the vitellogenesis pathway is used for vertical transmission. So organisms are either binding to vitellogenins or the vitellogenin receptor, and then using that to translocate and invade the developing oocyte. So we've been really focusing on this as a potential mechanism for vertical transmission. So this is oocyte development. This is a soft tick. The cuticle has been removed, exposing the fat bodies here, the midgut, and then ovaries. If we zoom in on the ovaries, what we show then are the hemolymph and the ovarian lumen, and then there are 5 stages of oocyte development. Stages 1 and 2 are arrested in female ticks that have just molted from nymphs and are virgins. So this is a pre-blood meal, pre-mating stage. They'll be in stages 1 and 2. Once that female tick mates or blood feeds, it can upregulate the vitellogenins, which are these little tan circles, and the vitellogenin receptor. The vitellogenin proteins will bind the receptor or translocated into the developing oocyte, and then they're broken down into smaller subunits that are then formed into larger yolk granules. And so this is where we think either through the vitellogenin or the vitellogenin receptor is where you can have transovarial transmission of a pathogen. So then at the end of stage 4, this oocyte is then translocated into the ovarian lumen where fertilization occurs. And then you have oviposition and embryogenesis. So let's talk about the vitellogenins specifically, and then we'll also mention the vitellogenin receptor, because there's actually been a lot of confusion in the literature with vitellogenins and another group of proteins, okay? So these are not easy. I think there are misannotations. So there's some limitations in annotations. Alex did a lot of manual labor digging and looking for these genes, and then we've done a lot of work validating them. And part of the reason is because vitellogenins are part of a heme lipoglycoprotein family. And this is just what the proteins look like with a signal peptide in these 3 domains. However, along with the heme lipoglycoprotein, in addition to the vitellogenins, are these carrier proteins. So this is what we've summarized about vitellogenin proteins. Their energy for yolk, for embryos, they make yolk. They're expressed only in females. They're not produced in male or nymphal ticks. Then they're detected at high levels in eggs. Carrier proteins really have nothing to do with reproduction. They're fat storage present in all sexes and all developmental stages, and they're low to undetectable in mature eggs. I'm going to show some data that we're trying to understand a little bit better, but those are like newly laid eggs. But in mature eggs, these are like, they become undetectable. So the first thing Alex did was when we identified some candidates, we called them putative vitellogenins and the putative vitellogenin receptor. And there were 6 of them, and here's the putative receptor. He isolated messenger RNA, did 5' 3' race to generate cDNA, sequenced it, and then translated it in silico. And from that, then we were able to generate a phylogenetic tree. So what this is just the PCR gel. We had bands at the expected molecular weights. And then what we started to do is then categorize them as carrier proteins or vitellogenins. So the red are the putative candidates, okay? So what I started renaming them then is vitellogenins, because they were grouping to vitellogenins of other argasid or exoded ticks. And then here were the putative carrier proteins that grouped with exoded carrier proteins. So we now started noticing this grouping. What we did then next is looked at an expression analysis. So what we expect with this is that they should be expressed in females. Blood feeding would upregulate the vitellogenins. What we used were female ticks that have been reproductively active, okay? So what that means is they would have all stages of oogenesis present. And what that also means is, like, we didn't think that we would notice a differential regulation in the vitellogenin receptor based on blood feeding in female ticks. And that's what we saw. So in these graphs, on the x-axis is an unfed female, unfed male, unfed nymph, and fed nymph. On the y is the target copies per 1,000 copies of beta-actin. And what we saw was a high upregulation of the vitellogenin 1 and 2 in fed female ticks. We did not detect expression in the males or nymphs, regardless if they were fed or not. I'm going to move this. So in the receptor, this was also consistent with what we thought. We did not see a differential regulation, again, because these ticks had all stages of oogenesis present. And so what we saw was in unfed females, fed females, expression of the vitellogenin receptor. And again, we did not detect expression in males or nymphal ticks. And then what we saw with the carrier proteins was that they were dependent on blood feeding. So males, nymphs, females, they all upregulated the carrier protein 1, 2, 3, and 4. There was no significant in some of the samples, but they were producing them at all stages, all sexes were producing them. So again, this was pretty consistent with what we were seeing. So the last set of experiments, then what we wanted to do was look at the protein level, So what Alex did was he took eggs when female ticks started to oviposit, he collected them shortly after oviposition. He also then took nymphs and he lanced their leg and then slowly centrifuged them to then collect the coxal fluid, flow through out of the tick, or not the coxal fluid, excuse me, the hemolymph from the tick. And so he took egg lysates, which are shown here, and the nymphal hemolymph, which we would not expect, vitellogenins, and ran them on an SDS page. He cut out high molecular, which are shown by these dots, these high molecular weight proteins, and then lower molecular weight proteins. And then this table are the proteomic results. We have the protein, peptide spectrum matches in eggs, hemolymph, unique peptides, eggs, and hemolymphs, and then the protein coverage. And so what we saw, we did not expect to detect much of the vitellogenin receptor in eggs, but we saw that vitellogenin 1 and 2 were highly abundant in eggs, and we did not really detect, if anything, in hemolymph. Carrier protein 1 was highly abundant in hemolymph. We did get some detection of that carrier protein in these eggs. So we're actually looking more into this. And then with the other carrier proteins, we did not detect them in the hemolymph. So what are we doing now? So we have this foundation, some ongoing studies is we're really interested in taking nymphs that molt into a virgin female and then looking again at expression of the vitellogenin, especially the vitellogenin receptor. I want to inseminate those so they're virgin females, inseminate them, and I think at that point we should see an upregulation in the vitellogenin receptor, and then see if that could clarify also some of the other with that carrier protein 1. But in general, I really think we've identified the genes for the vitellogenins. A phylogenetic analysis supported that. The expression analysis distinguished those vitellogenins from the carrier proteins. And then the proteomic analysis was validating at the protein level. So where we're going with this is we are really, really interested in this vitellogenesis pathway and transovarial transmission. We're using the Borrelia turicata model. So we've done a lot of genomics. We'd made a lot of genomic advances in Borrelia, now that we have the tick. And so it's almost like this idea has started 15 years ago, and we've just been really pushing the genomics resources, the genetic tools, and all of that to really now start studying the molecular mechanisms of vertical transmission. Pretty excited about that. But, again, none of this would have been done without a lot of help. All the work Alex did, Brittany, she's a staff scientist in the lab. I want to point out Michael, and then Serhi, he was the one that published transovarial transmission of Borrelia pathogens by autogeny. So I thought that we hadn't seen that in the literature for Ornithodoros turicata. None of this would have been possible without Pete Thiel and our collaboration for years. And then I can't express enough gratitude to the USDA. We tried different ways of getting this genome project going. It started us, and then we ran into walls, and when the USDA stepped in, that took us to another level with being able to do some of the long read sequencing and the HiC. And I'm just very grateful to Anna Perot. Anna, these are the people I really kind of have interacted with the most throughout this project. And it wouldn't have been possible to without the help and support of McKinsey, David, Shina, Brian Scott, and put her twice. This is that important. No, sorry about that. But yeah, so I'm just saying, like, really grateful for the USDA and the help that they have given. So with that, I will take questions.
All right, thank you, Job. Great presentation, and I do think Shenna is that important. She does need to be listed twice. So, so we have a good amount of like some time to close out the hour. So, first of all, have any questions that anyone may want to ask Job, and then we can roll into if you have any lingering questions from the previous three speakers. You're welcome to ask in. But I do have one for you, Job, which is so you don't see expression of any of the vitellogenin, not the receptors, but the other vitellogenins. You only see it in adult. Do you know if that is regulated at all by like early adulthood, late adulthood? It's like you just see expressions of that vitellogenin throughout adulthood.
Yeah, so I mean, with that study we used, like I kind of like mentioned in that, like reproductively active females. So we haven't looked at different stages of adulthood, but that's where we're going next, especially with I don't know, young adulthood with those virgin females, because it'll be interesting to see kind of like the dynamics or the kinetics of gene, activation with blood feeding. So we want to do that. So take virgin female ticks that had just molted and then blood feed them, and then take another cohort and then inseminate them and see how that impacts vitellogenesis, the expression of the vitellogenins. So yeah, we have not looked at different stages of adulthood or even late adulthood, right? Because these ticks can, they could go 5, 6, 7, 8 years. So that would be a whole other thing is looking at. So Pete Thiel, I want to say this, he has a cohort of ticks that have been that have been starved for years. So we've that could be also a possibility of trying to look at expression in like really aging female ticks.
And like, does mating have an effect on there? I guess I didn't realize, I didn't pay attention enough if that like if those were mated or unmated females, but like
those had been mated, yeah. Yes, they were just a cohort of and that's why we didn't see, I don't think, a differential expression with the receptor, because once they're mated, they will have all stages of oogenesis present. And then
that's what
and they don't need, I didn't really clarify this. So they can be inseminated one time and they will keep ovipositing. So they do not need multiple inseminations either. And I think that's really an interesting biological characteristic of them.
So they just store the sperm for like nearly indefinite amount of time. Wow.
So Job, I also had a question. You pointed at some early work on variation in the ability to transmit the swine fever. It seems like that's sort of pointing a generation of variants that maybe are correlated with transmission or not. So I'm curious if sort of it's old work and if you have sort of access to any of those old lines, or if anything is kind of ongoing that's going to try and replicate that.
Yeah, I mean, so I mean, it was interesting when I was doing this talk. I was searching on the internet and stuff. So it looks like there's a group at A&M that may be looking at vector competence with African swine fever virus. Is that what you're
kind of
like?
Precisely.
Yeah. And so I think something that would be really interesting, and I was thinking of reaching out to him, is like, because what we've done through the years is, and you saw that distribution map of turicata. So we've been trying to get different populations into colony, because in that paper, what they were looking at is they were looking at a population of tick from Kansas and then a population of tick from Florida. And they're technically the same species, but I think it's Dick Endritz that I don't think that's who. Don't quote me on that. But there was an entomologist, blanking out on his name unless it was Endritz, in Florida, that considered that Ornithodoros turicata population in Florida as a subspecies. And what we see in the lab in a different model with Borrelia, that there are definitely some differences in vector competence. So I think it is important to do with different different tick populations vector competence studies. That I think that definitely needs to be revisited, especially since now we have like some genomes and we can do more in-depth studies.
I
Yeah,
got
so maybe could we bring the whole panel in? Is that all right? Or Perot, what are you saying?
Oh, I just got one last private message, which was, and I think maybe they meant to add it. But so is there been any work to disrupt the vitellogenesis pathway that has resulted in like a control strategy before for any other species, or just?
Not in this tick. I think, oh man, was it Buophilis? I have come across some literature that they've tried to interrupt that pathway. So that's kind of something we're also interested in, like characterizing, is knocking down these genes and then seeing what happens with oviposition.
Cool. Yeah, so I guess if we can get all the other speakers, so if there's any follow-up question, and then while they're kind of turning on their cameras on, so we had a previous question asking about if for Michelle's talk, if the editor population was stable, do you need to keep releasing new populations? And so she answered it in the Q&A, which I guess I don't know if you want to vocalize that at all to help.
Yeah, I guess I might as well cover it again. So gene drive with insect releases, sort of the standard we compare to a lot of times is sterile insect technique, where you just release sterile males to try and collapse a population. And SIT takes releases of like, I think they're doing 7 times to 10 times a standing population. So if your gene drive can do something around like half the size of a population, it's generally considered to be more plausible than what people are currently doing. And then also in terms of doing multiple releases versus a single release, we assume that once you have a facility or a lab set up such that it can produce the transgenic individuals you want, it's easier to just continue producing that over the years as opposed to the amount of groundwork it took to get that facility set up in the first place. So those are the things we think about in terms of like long-term releases into the environment.
So Michelle, I had a follow-up on the sex determination stuff. So those modeling where you're manipulating sex determination and in the model are the drive, the editor, and the target both autosomal loci, or can you put them on sex
Autosomal
chromosomes,
loci.
and how does that change the dynamic?
I just looked at autosomal loci for those, but you could also, great question. You could also think about linking them to sex chromosomes, and I'm not sure how that would change, especially for looking at XY versus CW, but very interesting.
Because yeah, a lot of this group is clearly got to focus on the sex chromosome identification. So that's something that's always curious to me, is how we can change our approach if we can use different types of genome locations. So for instance, with Job talking about the vitellogenin protein, right? I'm sort of envisioning, okay, well, if there are vitellogenin alleles that are competent or that are not competent, right? You had this suggestion of sort of free adapting, or modifying, not suppressing a population, but sweeping a new allele through in a way that could, prepare us to not have transmissible ticks in the population. So yeah, we're close on the end. I don't know if I don't see any other questions coming in. Perot, did you have any other thoughts?
Nope, that's it. So I guess the last things to close out is just to say thank you to all our speakers. I thought you all did an amazing job, and thank you for understanding all of my massive emails, and thank you all for attending. So appreciate your time. Anything else I need to cover, Brad, or we got it all?
I think you got all, but one last thanks to Pro and David, the organizers for this session. A lot of work, a lot of time, a lot of effort, and all the speakers as well. Appreciate all your time put into it. Thank you very much.
Thanks all. Go get lunch or dinner. I don't know. What time frame are we at?